So thank you very much for the introduction. Thank you very much for attending this session. And thank you to the RSS for the invitation to attend and present this paper. So as introduced, my name is Claire Gormley. I'm a lecturer in statistics in the School of Mathematical Sciences in University College Dublin in Ireland. This was work with a former PhD student of mine, Gift Nyamundanda. He's now with the Institute of Cancer Research here in the UK. And also with a collaborator in UCD, Lorraine Brennan. So Lorraine is a lecturer in nutritional biochemistry, is her official title. So she's giving sort of the biological insight to this work. So the title of our paper is a dynamic probabilistic principal components model for the analysis of longitudinal metabolomics data. So there's a few different things in the talk. There's metabolomics data, which may not be something that's familiar to all of the audience. It's longitudinal in nature. We're using a version of principal components analysis, and the dynamic piece relates to the longitudinal aspect of the data. So I'll hopefully clarify each of these pieces by the end of the talk. So where this work originated was that Lorraine was conducting a study in UCD in the Conway Institute, which was a longitudinal metabolomic study of epilepsy. And she came to us with a number of questions, a number of things that she wanted to answer by collecting metabolomic data. We developed this method, which we've termed dynamic probabilistic principal components analysis, Dp Pca, so I'll give an overview of that. I'll talk through the application of our method to the actual data that were collected, and then I'll summarize and give some future directions. Okay, so the first part is to introduce the concept of metabolomics. So I'm very aware that there are people in the audience who are possibly a lot more knowledgeable about this area than I am, so please forgive my attempt as a statistician to describe a biological area. So most of us are familiar with genomics or proteomics. There's lots of new omics sciences around at the moment. The metabolomics view themselves as a slightly more global view. So genomics are our actual genes. Proteomics is the next level when genes tell us how to make our proteins, and metabolomics is a more global level again. So they would argue that it gives us a more complete view of the status of an organism or of a system. So what are metabolomics? Is a study of metabolites essentially, and what metabolites are small molecule products of reactions in our bodies, whether it's a human being or a plant or an animal. And the science then of metabolomics is simply the quantitative study of metabolites in a biofluid. It's a very nice science in that it's relatively cheap to run. We can get metabolomic data from a blood sample, from a tissue sample, from urine. It's quite relatively cheap in terms of the omic sciences. So many of us, possibly without knowing it, use everyday metabolite language or metabolomics language in our daily lives. By the end of this talk, you'll hopefully have lots more of this metabolite floating around your system, which is caffeine. Most of us are familiar with cholesterol, which is another metabolite. But then it gets more complex. There is metabolites. The one I'll discuss by the very end is called 2-oxoglutarate. So it gets more and more kind of scientific, I suppose. But they're things that we can, I suppose, identify with. And the interest of metabolomic scientists is that the patterns of these metabolites in our system, for example, can provide insight to the underlying mechanisms of disease. And this is the crux of the matter. The data are complex from a statistician's point of view. Metabolomic data are typically spectral data, so either collected from an NMR, a nuclear magnetic resonance spectroscopy machine, or from mass spectroscopy. So in Lorraine's lab in UCD, it's NMR spectra that she gives us. So in a sense, metabolomic data is simply NMR spectral data and is no different to any other context in which you'd collect such data. The issue, as usual, is that our number of samples and the number of people in the study is much smaller than the number of variables or the size of these spectra. So n is going to be the size of my sample, and p is going to be the number of variables in our study. So I say variables, Lorraine calls them metabolites. In terms of the spectra themselves, they're typically labeled spectral bins. Sometimes in the spectra, they're called peaks. So these 4 words essentially all mean the same thing depending on the field of science in which you're working. So I'll jump between them. The main problem being is that I have much fewer subjects than I have peaks in my spectra. The data are highly correlated. So metabolomic concentrations are very highly correlated. So if you have a high level of one metabolite, you might automatically have a high level of another metabolite. So we have correlation in that sense. Another issue is that one metabolite may be represented by more than one variable or by more than one peak. So in the NMR machine, a metabolite can sort of get split into 2 and appears at 2 different peaks. So we can't. While the metabolomic scientists are excellent at pinpointing that peak is the caffeine metabolite, we have to be aware that that caffeine metabolite might actually appear at 3 different peaks in our spectrum. So we have correlation within the spectrum. And then in the context of this particular study, as I mentioned, it's a longitudinal study, so we obviously have correlation across time. And then, additionally, lots of what is measured or shown to us in the spectrum is simply noise, and lots of the metabolites may not have any relationship whatsoever with what we're looking at. So the study itself that Lorraine developed in UCD was to essentially gain insight into the metabolomic changes, or the metabolic changes, that are going on in someone's system when they have epileptic seizures. When I say small n, or small sample size, I mean small, 15. I asked her had she dropped a 0 at the end, but she said no, it was definitely 15 subjects in the study. So we're dealing with a very small sample size here. 8 were treatment subjects and 7 control. Urine samples were taken from each of these 15 subjects over a period of 8 days. So in terms of longitudinal data, again, that's even quite small there. And essentially what Lorraine presented to me were NMR spectra that had 189 peaks. So for each of 8 days, I simply have she gave me a spreadsheet with 15 rows and 189 columns. So here's a picture of the spectra from a single time point, a single day. The black lines are the control subjects and the red are the epileptic subjects. And this is typically in the metabolomic literature how these spectra are graphically illustrated. So they never seem to have a y-axis, which as a statistician seems a little odd. But on the x-axis, or the horizontal axis here, we have the chemical shift, which is measured in parts per million. So each of these is a little peak, and as I mentioned, the metabolomic scientists can kind of allocate each of these peaks to a specific metabolite. So the y-axis, in a sense, or what should be here, is abundance. So a high peak means there's lots of that particular metabolite in that system. So there are essentially 15 lines overlaid here. They look quite similar, but you can see there are regions that there are differences. If you zoom in here, for example, we can see that there's definitely something subtly different going on here, and it's these types of differences that interest the metabolomic scientists. On the converse, I can think of these data as being measured across time. So on our x-axis here, I simply have the 8 days. And on the y-axis, I have the signal intensity, or the abundance, that y-axis. And this is for a particular chemical shift value, 2.46. So all I've done here is pulled out a peak around here for one control subject and one treatment subject, and shown what I'll call, in the language of the paper, the evolution of that metabolite across time. So you can see the abundance level of that particular metabolite changes across the 8 days. So Lorraine's, when she came to me with the data, I said to her, okay, nice data, but what do you need me to do? Give me your questions of interest. So she asked for 3 things. One was data visualization. So they like these metabolomic trajectories. They like to be able to sort of draw pictures of the movement of these subjects across time in a metabolomic space. She wanted me to assess the effect of time within each treatment group, so do things depend across time. And finally, to, as you would imagine, identify the metabolites which change over time within each treatment group. And most importantly, are those metabolites different between the epileptic group and the healthy group? So, to answer those 3 questions, we developed the Dp Pca model. If I start at the end, the PCA piece came from the fact, a bit like the previous speaker, that in the metabolomics world, PCA is the tool. They all understand PCA, they use it ubiquitously, probably too ubiquitously, but it's something with which they're very familiar, and they talk in that language. So that was the main reason for sticking with a PCA approach. As a statistician, I needed to extend it, so I reverted to a probabilistic version of it, which many of you may be familiar with, and then developed it further to this dynamic aspect. So to just really introduce some terminology or my notation, as I said, the standard approach in metabolomics is simply to PCA everything. So they apply PCA. Why? Because they project the NMR spectra at each time point into a lower dimensional space. So I'm going to call that lower dimensional Q, lower dimension Q. Typically in metabolomics literature, that's 2. Which reveals the inherent data structure. But they typically just sort of put all the data together from across time and PCA it. They then look at these metabolomic trajectories by simply plotting what we would call the principal component score over time for each subject. And then they use what most people would be familiar with the term loadings, the principal components loadings matrix, to identify the metabolites that are responsible for the data structure. So that would be their usual sort of steps in analysing such data. The limitations, obviously, are of kind of traditional PCA, is that it ignores the correlation due to the repeated measurements. As I say, they typically just push all the data together. It's not straightforward to extend the model, because there is no real model in standard PCA. And also, there's no sort of straightforward way of assessing uncertainty, let's say in the loadings or in the location of scores. So where did Dp Pca come from? There are a couple of other approaches in the literature to sort of further developing PCA to model longitudinal type data. I've given 2 references here, but both rely on kind of the traditional principal components analysis approach. Fittingly, back in 1999, in JRSSB, Tipping and Bishop introduced probabilistic principal components analysis, and really it's the fundamental basis on which this work is based. And essentially in that seminal paper, they showed that the maximum likelihood estimates from a Gaussian latent variable model are completely equivalent to the resulting loadings matrices and scores, for example, from standard PCA. So essentially what they did was they put PCA in a probabilistic setting, or they put a distribution on it in a way. So this makes things very nice. It gives us ways to address some of the limitations of PCA, because we now have a statistical model, a Gaussian latent variable model. So we can compare it to other models, we can do model extensions, we can do all of the things that we as statisticians like to do. Okay, so in one slide, what is Dp Pca? Essentially what we do is we model the NMR spectrum for subject I at time m. So it's just a vector of length 189, xim. And it's simply a linear function of Wm, which is going to be my loadings matrix at time m. The principal component score, you can think of it, at time m. So UIM here is going to be my latent scores plus some noise. So as I said, XIM is simply the data at time m for a particular subject. The loadings matrix is obviously going to be important. So it has P rows and Q columns. I'll talk about Q in a while. And the underlying assumptions of this probabilistic principal components model, first and foremost, are that these latent scores, or the principal component scores, follow a multivariate normal distribution, as do the error terms. In the Tipping and Bishop original paper, this was simply an identity covariance matrix, and this was just sigma squared I, because they had no time dependency. So for the purposes of our model, to model the fact that we have a longitudinal aspect to the data, we now have a sigma squared m as the covariance of the errors, denoting each of the 8 time points. And for the covariance of the scores, we no longer restrict it to be the identity. It is a diagonal covariance matrix, but those values are not necessarily constrained to be 1, and we allow a different covariance matrix at each time point. So we very simply model the potential correlation or dependency across time by putting a stochastic volatility model, sort of embed that in the dynamic principle, or in the PPCA model. How do we do that? We simply put the variance of these latent scores using a stochastic volatility model, and likewise the variances of the errors. It's probably easier to go back to this slide and say essentially what we do is we simply put a stochastic volatility model into both of these parts of the standard PPCA. To get slightly more technical, we actually put a stochastic volatility model on the log of those variances. So for the rest of the talk, I'll call these log volatilities. And this is really a herald or harks to the literature in which these stochastic volatility models are mostly used, which is the financial literature. And there they talk about log of variances or log volatilities. So my language somewhat changes. So essentially what we do is we take the log of those variances and we model those. We simply model them using an autoregressive model of order 1. So I say my log volatility is equal to some mean plus some persistence parameter phi, which depends on the log volatility from the previous time point. So this is for the log volatilities for the principal component scores, and likewise a very similar model for the log volatilities of the errors. We have mean parameters which aren't really of interest to us. What we are particularly interested in are going to be these persistence parameters. So the phi parameter here tells us about the sort of the degree of dependence between time point m-1 and time m. So they're going to be of interest. And then again, showing the literature, they call these error terms, the R's here, innovations. So for brevity, because there's a lot of terminology here, I'm just going to really call these theta 1 and theta 2. Just think of those as being the stochastic volatility parameters. Okay, so back to Lorraine sitting in my office and saying, what do you want me to answer for you? What are your questions of interest? So her first question was about the data visualization. So to answer that part for her, you can think of it as those U parameters, those latent principal component scores. So I now have one of those for each subject I at time m. So I'm going to use those to answer that part. Her second question was, what's the effect of time? The parameters that answer that are those phi persistence parameters that talk about the dependency between time m-1 and time m. And our final question was, which metabolites evolve over time and which are different between the control and the treatment group? And that's simply, let's look at each loadings matrix, which I call W, at each time point m. So it ends up, we're interested in sort of 4 different parameters. Keep this light. We used Bayesian Inference to examine the parameters of the dynamic principal components analysis model. It makes things nice and easy. The augmented likelihood function looks a little messy, but it's actually straightforward. So I've only one slide really on inference. The augmented likelihood is going to be the density of the observed data, the latent scores, the latent log volatilities, given some parameters. So we have our m time points, so conditional on the latent scores. Essentially, we can model these as independent. I'm assuming independence across my n subjects. This is the model for our data. I have the model for our latent scores, the model for the log volatilities, and the model for the other part of the log volatilities. It looks complex, but every single one of these densities are simply multivariate normal Gaussians, so it's quite straightforward. We simply use conjugate priors, and I use a metropolis within Gibbs sampler. So just to bring the talk towards its end, we then apply this method to the actual data, and what did we find? So first problem was the data visualization. So here's an example of 2 metabolomic trajectories. They wanted to be able to visualize how these subjects move across time. Black is control, red is treatment or have epilepsy. This is principal component 1, and this is principal component 2. The digits in the plot represent the different days. So we start at day 1 and we move to day 8. What we can clearly see is that the variation, or the sort of the spread of movement of someone with epilepsy of their metabolomic system is much more variable than that of a control patient. Some of you may be preempting a question saying, well, which principal components are they? These are the principal components of the first time point, PC1 and PC2, and what we do is we rotate using kind of Procrustean techniques, the latent scores of the later time points to match those. And I can discuss that further if needed. Second thing was to assess the effect of time within each group. So that were those Phi parameters, if you remember, they were the things that we were interested in. Here we fitted the model to each group separately. And what we end up with is, for example, this is the posterior distribution for the Phi parameter in the epileptic group. So you can see the posterior mean was 0.69. You can see there's quite a high level of dependency as we move across time. Within the control group, it was slightly lower, but the same type of dependency seems to be there. And then finally, to identify the metabolites which evolve within each group. Here we were going to look at the loadings matrices at each time. It's a little bit messy here, but we simply looked at principal component 1 in each of these 8 loadings matrices. We ranked the metabolites in each one by the magnitude of their loading and pulled out the top 5 at each time point. So, for example, at time point 1, we pulled out the top 5, what I'll call influential spectral bins. That just means they have the largest loading on PC1. And what we get, which is much nicer than sort of standard PCA, we still get loadings. So, this is the size of the loading on variable 2.54, but we also get uncertainty bars around those. So, for example, in the epileptic group, we were left with a total of 8 unique spectral bins when we looked at these top 5 across all 8 time points. So we call these our 8 influential bins. What we wanted to do is look at which of these evolve over time. So we did a very straightforward linear mix model to the data associated with those 8 spectral bins. It's quite straightforward. We simply did a cubic time effect was our most complex model and pruned backwards. And in the epileptic group, it turned out that of the influential 8 spectral bins, 6 of them were deemed to fluctuate or evolve over time. In the control group, there were 7 influential spectral bins, and all 7 were deemed to evolve over time. So picture will hopefully explain this a little better. Left is the epileptic group. So on the x-axis, I have the 8 time points. The y-axis is the predicted average intensity under the linear mix model. So you can see how some of these metabolites evolve across time. What's the difference between A and B? So the one thing that Lorraine immediately spotted was if I focus on this metabolite known as 2-oxoglutarate, which is represented by 2 spectral bins, 2.46 and 3.02. So 2.46 is the black line and 3.02 is the blue dashed line, and the same in the right-hand plot. So I'll drop everything else, all of those other curves, and just focus on those, and you can immediately see a difference in the way this metabolite evolves across time between the epileptic group and between the control group. There's very different quadratic effects going on there. And this was something that they hadn't sort of identified before, and they're now exploring it further. I did some posterior predictive model checking on the bequest of the nice reviewers from when we submitted it to the JRSS. We had omitted it in our initial submission. So how do you assess model fit when it comes to multivariate longitudinal data? So what we did in the end was we simulated data from the posterior predictive distribution, nice and straightforward within the Bayesian framework. And we simply looked at the mean absolute deviation between the observed data covariance matrix and the covariance matrix of each simulated data set. And what we did then were these mean absolute deviations, the MADs. We simply looked at a histogram of these values, and you can see for by and large, they're all quite small, hopefully suggesting that our model fits reasonably well. So to conclude, Dp Pca is, I feel it's a little bit of a cheat. Essentially, it takes Tipping and Bishop's PPCA model and extends it in a very straightforward manner. It does so by combining PPCA and stochastic volatility models, and this allows us to disentangle the different types of variation or of dependence, essentially, in longitudinal and high-dimensional data. The Bayesian Framework is particularly nice. It gives us nice ways of assessing uncertainty, gives us full posterior distributions, and so on. In the terms of the context in which this data was applied, it gives us a nice way to be able to visualize these metabolomic trajectories in sort of a principled way. It allows us to estimate the longitudinal effect of a study, and this sort of two-part step using the Dp Pca model, followed by some simple linear mixed models. In a sense, you can think of this as the Dp Pca does a bit of sort of variable selection, which are the important variables, and then we use standard linear mixed models to analyze them. And this gives us a panel of influential variables which evolve over time. So I think I'm way ahead of time. But just to conclude then, so just some references. This is our paper, I think soon to appear in JRSSC. I briefly mentioned in the talk that a lot of this stochastic volatility work is done in the more financial literature. So the Dp Pca model is similar but subtly different to a model of Aguilar and West from the Journal of Business and Economic Statistics from 2000. We have another paper that Gift worked on that works in a related area, and I'm sure most of you know it, but the seminal paper of Tipping at Bishop. Thank you.
Thank you, Claire. Sorry. We've time for a couple of questions. That's perfect timing. So I would have asked you exactly the same thing. How do you check those models? Because they are extremely...
Were you the reviewer?
I wasn't, but I might have been. And my other thought was, how do you set up the prior distributions for those? Some of those parameters are very difficult to get some kind of intuition on what they might look like. I just wondered how you were quite quick about that.
Yeah, so I'll jump back to some valid questions. Yes, so I place conjugate priors on the loadings matrices. So it's actually quite light on parameters. It looks like quite a complex model, but there's actually quite a few parameters. We have each loadings matrix. So here I simply put a normal prior on each entry in the loadings matrix, centered around 0 with large-ish variance, so quite an uninformative prior there. But hidden in these θ1 and θ2s, all right, things get a little bit tricky. So if I focus on θ1 actually, because these are the parameters of this autoregressive model. So this is just a mean parameter, so I just put a normal prior there. It's, again, relatively uninformative. For the variances, I think it was an inverse gamma, which again is sort of relatively standard and non-informative. The Phi parameter, this persistence parameter, is where things get tricky. There's a good bit of literature, again, in the financial arena mostly about the different priors and their different effects on this parameter. We did some sensitivity analysis that is included in the paper, and we looked at using a beta distribution, but what we actually found it to be quite sensitive to the hyperparameters that you include in if you use a beta. For us, we used a truncated Gaussian between plus and minus 1 with a very flat variance, and found it to be much less sensitive. The results of the model seem to be much less sensitive to that. But this Phi parameter, and likewise here, that's not without its difficulties.
Okay. Any other questions? Wait.
Sorry, my question might not really be about your talk, but I was wondering, in these spectra, is the x-axis meaningful? So, because I think the basic method you're using assumes a general correlation structure. Would you not expect some autocorrelation across the x-axis?
My understanding of the spectra is that there's no sort of meaning as you move from left to right. As I say, the peaks can be correlated. So if you have a peak at spectral bin 2.46, you're going to have a peak at spectral bin 3.02, depending on certain metabolites. But bar that dependence, there's nothing else sort of as you move across.
So these are discrete time models. So it's day by day by day. How sensitive is it to exact measurements at the same time of day, and is that controlled for it all? Or?
Very sensitive, and I'll be completely honest and say these weren't 8 consecutive days. So there was obviously a much larger data set, but with huge holes of missing data, which is kind of the next add-on, which is relatively straightforward to do within the Bayesian context, is sort of impute that data. But what we ended up doing was just pruning it back to what is the smallest number of complete times we have. So I definitely think there's sensitivity there to the the time of day and even the gaps in the measurement across time.
Okay. Thank you. So can we, I think we've had 3 great talks. I don't think anybody's told the climate people that Sheffield's warming up, because it's absolutely freezing in here. So you all want to go and get some coffee. So can we all just finally thank all 3 speakers for their contributions today? Thanks.